Autophagy dysfunction represents a hidden yet powerful link between two of the most devastating outcomes of aging: neurodegenerative diseases and cancer. Though these conditions may appear to occupy opposite ends of the biological spectrum, one marked by degeneration and the other by uncontrolled growth, they both arise from a failure of the body's core protective mechanisms. Chief among these is autophagy, the cellular process responsible for identifying, isolating, and digesting damaged proteins, organelles, and other toxic debris. When autophagy falters, particularly in aging tissues, it opens the door to a cascade of dysfunctions that promote either neuronal loss or malignant transformation, depending on the context and cellular environment. In the aging brain, autophagy plays an essential role in maintaining neuronal health. Neurons are long-lived, non-dividing cells that rely heavily on autophagy to manage protein quality and mitochondrial health. They are especially vulnerable to the buildup of misfolded proteins and defective organelles, which, if not cleared, can form toxic aggregates. In Alzheimer's disease, for example, impaired autophagy contributes to the accumulation of beta-amyloid plaques and tau tangles, hallmarks of the disease that disrupt synaptic function and eventually lead to cognitive decline. Similar mechanisms are implicated in Parkinson's and Huntington's diseases, where dysfunctional autophagic flux fails to remove toxic proteins like alpha-synuclein and huntingtin. These pathological accumulations are not just passive consequences of disease. They actively drive neurodegeneration, inflammation, and cellular death. The breakdown of autophagy in the aging brain is multifactorial. Lysosomal function declines with age, reducing the cell's ability to degrade autophagic cargo. Regulatory proteins like Beclin 1 and LC3 are expressed at lower levels. Mitochondrial damage accumulates, fueling oxidative stress and further impairing autophagy. This creates a dangerous feedback loop. The more damaged components that accumulate, the harder it becomes for neurons to maintain their function, and the more likely they are to die. The result is a slow but inexorable loss of cognitive and motor abilities, a descent driven in part by the quiet collapse of cellular self-cleaning. In cancer, the role of autophagy is more ambiguous but no less critical. In early stages of tumor development, autophagy acts as a suppressor. It clears out damaged DNA, oncogenic proteins, and defective mitochondria that could otherwise trigger malignant transformation. But in established tumors, particularly those in nutrient-deprived or hypoxic environments, cancer cells often hijack autophagy to survive. This allows them to recycle intracellular material, resist therapy, and thrive under metabolic stress. The same pathways that protect the brain from neurodegeneration can, in another context, support the survival of dangerous tumor cells. This paradox, that the same autophagy dysfunction can promote degeneration in neurons and hypersurvival in tumor cells, underscores the complexity and context dependence of the system. In neurons, too little autophagy leads to toxic accumulation and death. In cancer cells, too much autophagy can support survival and resistance. The warning here is that any effort to target autophagy for therapeutic purposes must be highly specific and timed with great care. Stimulating autophagy to slow cognitive decline may benefit the brain, but if undiagnosed tumors are present, this same stimulation could strengthen the very disease it aims to prevent. Conversely, inhibiting autophagy to weaken tumors might impair the brain's ability to defend itself against degeneration. Still, the overlapping mechanisms of autophagy dysfunction in neurodegeneration and cancer offer a promising avenue for research. Biomarkers of autophagic flux may serve as early indicators of both cognitive decline and cancer susceptibility. Therapies that restore autophagic balance, rather than simply activating or suppressing it, could be designed to selectively target vulnerable tissues. Compounds like spermidine, resveratrol, and NAD+ boosters are already being studied for their ability to support healthy autophagy without overstimulation. Dietary and lifestyle interventions, such as intermittent fasting and exercise, offer additional ways to support brain health while potentially reducing cancer risk. Ultimately, autophagy dysfunction is not just a cellular inconvenience. It is a central feature of aging that shapes the trajectory of multiple diseases. Whether a cell degenerates or becomes malignant may depend less on the presence of mutations and more on the status of its internal quality control. In this light, Maintaining autophagy through midlife and beyond becomes not just a strategy for preventing neurodegeneration or cancer individually, but a broader approach to preserving the integrity of the aging body as a whole. This convergence of pathology, where the same broken machinery leads to either mental decline or malignant growth, reveals both the vulnerability and the opportunity of aging biology. If we can keep the cell's cleanup crew functioning, we may be able to delay or even prevent the worst consequences of aging. But if we ignore it, the fallout may be far more complex than previously imagined, simultaneously eroding the brain while fueling hidden threats elsewhere in the body.